Thank you for coming to this panel. So we just ended a rather important panel on ethics, and maybe it's a good segue to talk about the importance of making drug product and talk about the field of innovation. This topic with regards to manufacturing, I think over the years everyone has always mentioned the products to process and the processes to product. For many of you who've been in the field for quite a long time, we're going to talk about your views on the next generation of technologies and future innovations in the field. Maybe one by one, if you can briefly introduce yourselves and what group that you're with, if you want to start, PJ.
Yes, hi, I'm P.J. Brooks. I'm the Deputy Director of the Division of Rare Disease Research Innovation at NCATS, National Center for Advancing Translational Sciences, one of the 27 institutes and centers of the National Institutes of Health.
Shawdee.
Thank you. My name is Shawdee Eshghi, and I lead the cell therapy and gene editing work at Ginkgo Bioworks. Ginkgo Bioworks is a horizontal platform for cell programming across industrial products, including therapeutics.
Ken.
Yeah, hi everybody. My name is Ken Prentice. I'm the Vice President of Process and Product Development at Shape Therapeutics. Shape Therapeutics is an RNA editing company with technology pillars across RNA editing, AAV capsid engineering, and manufacturing, AAV manufacturing.
And Mercedes.
So hi, my name is Mercedes Segura. I'm Vice President of Process Development at ElevateBio. ElevateBio is a cell and gene therapy company. We develop our own therapeutics with our own innovative technologies, and we support the development of others that we partner with. So it's a great pleasure to be here.
Thank you. And I'll try my best to moderate this panel today. Raj Prabhakar, I'm the Chief Business Officer at Rocket Pharma. We'll be presenting tomorrow, but for those of you who may not be familiar, we're a multi-platform gene therapy company with 6 disclosed programs, 3 in the AAV space and 3 in the lenti space. And I've been in the field for more than 20 years, more than half my career in cell therapy. So maybe I'll start with the question. What drew you to this field? Maybe, coming, let's say, out of grad school, or maybe talk about how you pivoted to get into the cell and gene therapy space, and talk about what drives you to stay in this field. Maybe if you want to start.
Sure. So I started out as a basic neuroscientist in various career twists and turns. I ended up working with a rare genetic disease. I ended up meeting some of the patients with that disease, and it really stuck with me, the idea of what is my research really doing to help these people. And around that time, gene therapy was kind of making a comeback. The discovery of AAV that could cross the blood-brain barrier was made, and I realized that for many of these diseases, you can almost think of all monogenic diseases as, in a gross sort of way, like one disease. The problem's the same, and the solution's the same. You need to get a working copy of the right gene into the right cell at the right time. And if you can do that, that's a broad approach to many rare diseases. But then also the realization that some diseases get these treatments and they're amazing to see, but there are other diseases that could equally benefit from them that won't, because they're not big enough to be of commercial interest. And that led me in many ways to the position I have now in many of the programs that I'm involved with, and I can talk about that later.
I think for me, I'm really passionate about technology development. I'm a chemical engineer by training, and it's been a real treat and honor to be able to invent new technologies that will lead to completely new therapeutic modalities. In my previous job, I worked at a gene editing company, and I remember the day that we got iPSCs in-house, and having a freezer full of Cas9 and a vial of iPSCs just really felt like anything is possible. And so that's something that we bring to our work at Ginkgo, where we're technology agnostic and really want to bring the best solution to solve the challenges across discovery or manufacturing. And so it's really great to be up here with other people who are building similar technologies in the service of patients.
Yeah, I guess I like that comment earlier. My career has taken twists and turns to put me in this seat today. For me, the first half of my career was in biologics space, working for a large company in the Seattle area on monoclonal antibodies. And when I joined that space, it's almost like a case of deja vu. We had very poor performing processes, struggling to meet demand for a blockbuster therapeutic at the time, and I got to live through the building of a platform. And that's how I grew up, was really living through that antibody platform building. And so unfortunately, the tail kind of turns a little bit tough. They close the site, and for various reasons, I need to stay where I was. But there was a small company that was growing up at the time called Therapeutics at the forefront of autologous cell therapy. I didn't know anything about cells. I was, at the time, doing kind of esoteric mass spec work in a lab, but somehow got on with that company and, again, was able to be a part of sort of this transformative therapy that came along for the ride with process development and understanding how do you control a system in order to make something. And so when I got the opportunity to move over to Shape Therapeutics, it was really this convergence for me of a modality that seemed to be one that I could really wrap my head around, a need from a patient perspective, and a driving technology that was really exciting. And so the thing that excites me is that convergence. It seems like over the last couple of years, we've gotten this hook for gene therapies that allows us to turn the corner a little bit and to be a part of that and to be on the leading edge of that. It feels like a one-in-a-lifetime kind of opportunity to be involved and invested and watch the industry grow. It's a very exciting time.
So for me, the story started in 2001. I was looking for a good place and a good topic to do my PhD, and I came across a group in Canada, and they offered me, okay, you're going to do a thesis on, vector for gene therapy. This was 2001. And at the time, it sounded like science fiction. I didn't know if to believe it or not, but I did, and I moved to Canada specifically to do that PhD. And the concept was very simple. It seemed like something that we could do. There was a broken gene. We put the right gene in, it fixed, and it sounded like super good. So I moved to Canada to do my PhD, and then I stayed a number of years in academia, and finally I moved to the industry with Bluebird in 2013, and AgroBio after, and then ElevateBio. So I've been in cell and gene therapy for 23 years now.
So it's great to see your firsthand experience in areas between cell therapy, gene therapy, CAR-T over the last 20 to 25 years. Maybe let's pivot the discussion a little bit. The next 3 to 5 years, what is the technology where do you think the innovation is going to come from? What is the thing that gets you really excited? And maybe even putting in context, something that can help treat a broader number of patients than the current scalability. Maybe we'll start in reverse, Mercedes.
All right. Yeah. So I truly believe the first, these past 10 years were establishing proof of concept, right? Coming up with the first industrialized processes, right? That you could develop a therapeutic that you could commercialize. Prior to that, it was all academic. And I think it's very young, the field is very young, there's a lot to do, and particularly in technology, right? Technology for processing, process intensification, process integration, simplifying the processes. The therapeutics are very complex, and they're not getting any simpler. So the manufacturing processes and the understanding of the product, the analytics that go with it, are going to be complex. So we need to be prepared for that challenge in order to master that and be able to develop the therapeutics in a way that people are looking for. So in every single one of the talks, I heard cost, right? So yes, I think monoclonal antibodies 30 years ago were in the same boat. And it's going to take time, investment, and effort to develop these processes and get the understanding of the products to the level we need to have them in order to have it accessible for everybody.
Yeah, I think, this is a CMC talk, but maybe I'll go outside of that for a little bit and then come back. But I think right now, it wouldn't be a surprise to anybody to say that delivery is the thing that I'm most excited about. I think in AAV specifically, that's what we work in. It feels like that the wild type serotypes have had their day. And now really looking forward to engineered or novel capsid serotypes to be able to deliver the right therapeutic to the right tissue in the right dose amounts. And I think people are doing some very exciting work on that, including us. I mean, we've got a AI-driven discovery engine that we're excited that's tuned towards that capsid discovery. So that's kind of outside of CMC. On the CMC side of things, I totally agree with Mercedes. Titer is a challenge. I referred to my antibody experience when I joined that. We couldn't make enough to meet demand, and that's where we are today in the CMC space for gene therapy. And so, the industry is maturing. We're understanding what we can get out of cells, how we can make them work harder. I think, quick back of the envelope calculation, folks would be surprised at how lazy our cells are today. I think there's a lot of room that we can make there. And because we're all working on platforms, we're all kind of, it feels like converging on similar ways to make these therapeutics, we have the opportunity then to drive farther and faster. And I think we're at that inflection point. I'm excited to see the vendors out, the consumables manufacturers folks thinking not only large but small. I think the N of one patient is out there, and while we can work our side from a production standpoint, we need the industry to come along for the ride with us on the consumable side, on the instruments and whatnot that can get us out to a clinical space. So lots of excitement.
And maybe just a subsequent question. You mentioned delivery, and I think earlier you mentioned like IV delivery. So do you think there's innovation on the device side potentially for, let's say, catheters, different routes of administration, different organs where you get tropism? Any thoughts around that? Do you see innovation there happening?
Yeah, I don't know that I'm the best to talk about that, being a CMC guy, but I do think that right now it's very challenging to have to pick, between a local dosing and a systemic dosing, and how do you find that balance? And so I think there absolutely is a lot of space and a lot of room for novelty in delivery.
Thanks. I think what I would add to the experience of Mercedes and Ken here is that I think that we're in a space where discovery can actually be part of manufacturing improvements, right? Our ability to screen biology at larger scale and come up with an optimized product may actually lessen the manufacturing burden down the line. So for example, maybe part of the AAV manufacturing challenge is that the capsids are unoptimized and the promoters aren't strong, and the genes are getting silenced, right? And so it's easier to screen a larger amount of space now than it was maybe, like 10, 20 years ago. And so that's a lot of the work that we do at Ginkgo, both in actually improving manufacturing platforms, but also in helping our partners with their discovery process with an eye towards what is it going to be like for manufacturing. So for example, we're heavily invested in circular RNA right now, and that includes both looking at diverse genetic elements to drive circularization and translation, but also thinking about how are you actually going to make this, right? And having platforms for both high throughput as well as high yield production. And so I think sort of closing that loop between discovery and manufacturing as early as possible in the development of a new therapeutic modality will certainly make some of the CMC challenges a little easier down the line, or that's the hope.
I guess I would agree with Ken about delivery, and I think We're all familiar with AAV and the pros and cons. I'm involved in a variety of AAV projects, but I think looking to the future, I'm sort of most excited about better ways to deliver gene therapy and also particular gene editing to cells and tissues other than the liver. Everything seems to go to the liver, and that's actually a problem. And so as part of the NIH Common Fund Genome Editing Consortium, we've funded a lot of work on better delivery systems, and right now we actually have a prize competition open for better ways to deliver genome editors, either to target them to specific cells and tissues, like sort of like a zip coding approach, and also for a non-viral delivery system that would cross the blood-brain barrier. And that competition is still open if anybody's interested. It's not a typical NIH grant, it's actually a prize competition.
It's an interesting segue to the next question. Trying to create competition to see what kind of winners or new innovation would emerge. So maybe what other approaches could you say could accelerate the development in CMC or technology? What are the other things that maybe that's not getting done or could be done in a more aggressive manner?
Somebody else can
Anyone
start.
could start.
I'll jump in there. I think really having a basis for your platform for gene therapies is incredibly important. Having that, again, I'm biased on the CMC or process development side of things, but if you can make the same thing over and over again and have very little variability between that, then all of a sudden you can start asking the question of why is it working that way? Can I perturb the system in order to make it better? Can I add an element here to make it express higher or lower? And I think the industry, until very recently, really struggled with that. There was a paper that, I can't remember the date now, but it's been a few years, where they just sent AAV out to a number of different organizations to measure how much was there, and it was like a log-fold difference across those labs, even in labs that were using what they thought were the exact same protocols, and that's a huge problem. I mean, at that point, we don't even know what our dose is that we're putting into people because we don't know how we're measuring it, and I think it's a little bit tongue-in-cheek there. I'm not trying to throw anybody under the bus, but it's a huge problem. And so I think one of the things that we have done internal to Shape is we've really focused our kind of core technology of editing and looked at can we make or how to make a stable producer cell line for AAV, and we've been successful in that, and that is the bedrock around which then we can build off of, and we've been able to leverage that and ask those questions of, how are things assembling? Why are they assembling that way? What in the process? What levers do we have to be able to push titers higher or packaging higher, or any of these product qualities that are pertinent to AAV? And I think that is something across the industry that really we can do is find that bedrock to all have a common element so that we can push farther and push higher. Otherwise, we're all kind of stumbling around wondering, well, did I make what you made, or did you make something different? And it's really hard to do an apples-to-apples comparison and then know where you are to go farther.
Yeah. I think in the next years we're going to see, what we've been seeing so far, that is, pushing the field forward, partnering, right? There is more data sharing, more partnering, more of those things that will help, share the information, get standardized procedures, standardized assays. 10 years ago, you would have to develop everything yourself, right? Think about, these are very complex products. You have a plasmid, you have a vector, you have maybe a gene edit, you have a cell. You can't do it all, right? Develop assays for all of these, acquire assays for deep understanding of all of them, and then expect to have the best processes for all of these different type of biologicals that give you just one product, right? So you really need to find suitable partnerships and leverage each other's knowledge in order to, and come up with standardization and, regulations that are going to help drive the field towards more standardized ways of doing things. And then there is the technologies. There is a lot of technological advancement going on, a lot of new instruments that come and solve the problems that we've seen. Part of the issue is people don't adopt them fast enough, right? They don't want to change everything in the middle of clinical trial, and part of the reason is they don't really understand the product, right? So we need better analytics to really understand the product, how the product, mediates safety and efficacy. Do we really understand that? If we were to understand that very carefully, we could make more changes. But it's hard to pass on innovative solutions when you're in the middle of a clinical trial. So that's part of the limitation, I think.
I think I would build on that by saying that our ability to do larger scale experiments has just exponentially increased. And so screening more clones, screening more plasmids, collecting more data is just faster and easier now. And so I think it should be easier to take a more optimized process forward in the same amount of time, together with not just advances in lab automation, but also bringing AI in to sort of accelerate some of the work that to make sense of all the data that we've already collected. So again, the clones for producer cells is one, but adding in like host cell modifications and plasmid refactoring to really bring sort of all of the pieces together for a particular process. I think that's something where Mercedes' point, like everyone would just sort of do one piece of it by themselves or attempt to do all pieces. But there are ways to kind of outsource or standardize parts.
Yeah, I'd just say one of the programs I'm involved with is something called the Bespoke Gene Therapy Consortium, or the BGTC. This is a public-private partnership organized by the Foundation for the NIH and includes many private sector partners, many of whom are here, including our host, the Alliance for Regenerative Medicine, many parts of the NIH and the FDA Center for Biologics, and Dr. Peter Marks. And one of the goals of that program is to develop a streamlined and standardized approach to get from the idea for an AAV gene therapy all the way up through a clinical trial. And one of the things that we're doing there is to see if we could identify a standardized minimal set of critical quality attributes that could be used to assess whether an AAV prep is safe enough to put into human beings. And so we plan to develop that as part of our IND package and ultimately make that available into the public. And there are a lot of other that involve data sharing within the BGTC, and I think there's a real opportunity there for data sharing to make some progress in some of these CMC issues.
So many of you worked in the AAV space, and let's say you've also worked in a suspension platform. So let me ask, and for many of those here that are new to this space, let's say that in your manufacturing facility you have an upstream room, and that whole process up to transfection is somewhere between 3 to 4 weeks. Then you have a downstream room that could be another week where you have purification and trying to get as much DNA in the capsids, and then you have your fill finish, and the analytics, as you mentioned, is complex. So the total cycle time could be something like 60 days. There's a lot of moving parts, and those of you sat in on some of who are very familiar with the nuances. The bill of materials to make a drug product could be over 100 to 120 distinct items when you include the consumables, all the raw materials, buffers. What are some of the biggest pain points from your perspective? Where is the biggest inefficiency? You can't improve everything. So if you have to solve some of the biggest problems, let's say in upstream or downstream or analytics, where is like the highest cost driver or the biggest concern, especially as you start to get to larger manufacturing?
Yeah, I mean, it's AAV, but Lenti, the cell therapies, they all have same common features, which is very complex and long processes. So if to any extent you can integrate unit operations, create less unit operations and achieve the same thing, you'll be saving time and cost. I do think we need to simplify the way we produce these therapeutics at one point and become more efficient, and drastically change things. So with advanced therapies, we're not talking about a twofold improvement. We need hundreds of fold improvement, right? So how are we going to get there doing the same thing, or, you know, with small incremental changes? Not that there's going to be a magic bullet that one thing is going to change everything, but we're going to take more risk and make more drastic changes in the way we do things, more creatively. Yeah, that's what I think. Eventually, I think we're going to come with better ways of producing these therapeutics.
Yeah, I guess I would say that right now in AAV, my perspective is tighter is king.
Tighter is king. Yeah.
Tighter is king. Yeah. We're not making enough, and so no matter how much work we do on purification, how much formulation, any of that, it's not going to solve the production problem. Everything has to be done on the upstream side of things, and so. To the point we have to do better. We have to make more, and then that'll allow us to once we make enough material, all of a sudden gaining efficiencies off of that process becomes easier. Whether it's an economy of scale or just an economy of kind of dose per unit volume. I think, this morning there were some numbers that were thrown around and comment about how, it felt like the medical industry could handle the cost of gene therapies. And it sort of made me sad when we were talking about, thousands of liters treating hundreds of patients. Because I think if you look to other biologic sectors, that number is completely inverted. I think you're talking about millions of patients out of thousands of liters, and small molecules is even bigger than that. And so we got a long ways to go, and really, our focus is on, how do we best produce the material, and then we'll let the other parts kind of come along for the ride as we make progress there.
Go ahead.
I'll just come back to the discovery is also part of a manufacturing platform where, for example, in our CAR T work, we've optimized the intracellular signaling domains of the CAR, where we get far greater proliferation of our T cells than a standard 4-1BB CAR. So that means with a smaller starting dose, you could achieve the same amount of killing and cytokine secretion. So just one example of, obviously a preclinical discovery here, but one example of how, putting in the screening effort to design an optimized product could lead to lessening the manufacturing burden. Similarly with some of the AAV work we've done in cell type specific promoters, if you get the expression where you need it, and maybe not in the liver, right, then maybe that'll also lessen the tighter demand at the other end. So I think that I've seen enough examples of how for our partners' products, the discovery can actually impact their manufacturing that it seems like the innovation should go together, right? And in doing all the work on the manufacturing platforms, but also trying to bring the most optimized products, nominating the most optimized products to that stage.
I got nothing to add on that one.
No innovation at the NIH.
Oh, no.
No inefficiencies at the end.
No, I don't have the direct experience working in this area to answer.
Mm-hmm. So maybe let me ask, from your vantage point, seeing what industry does, a large industries, where do you see inefficiencies, or where, if you looked at from a distance, where could things improve?
I'm going to give a rather different answer than you might think. I think the problem is really is thinking about developing treatments for one disease at a time, and taking what are really therapeutic platforms and not using them as therapeutic platforms, using them to make treatments for one disease. I mean, genome editing, if that's not a therapeutic platform, I don't understand the term. But we take that technology and we use it to treat one disease at a time. And not only does it not make scientific sense, it's a problem because if we do that, we're never going to get to all the disease that can benefit from it, the ones that are so rare. So what I would love to see is for develop either AAV gene therapy or more likely gene editing as a therapeutic platform from the beginning. Say one delivery system, one editor, more than one disease. And we did, in fact, as part of the genome editing program I mentioned, we actually funded one project to do that, to kind of demonstrate, I think, the way that would make a lot more sense scientifically and the way we could get to a lot more patients. I realize that does not seem to make business sense, and I don't understand why. I'm not a business guy. But scientifically, and if we want to get to the most patients as possible, I think that's the way we need to do it.
So ideally, if the platform works for disease X in CNS, you would change the gene of interest plasmid, and hypothetically, it could work for other CNS diseases with a different gene of interest. So if the agency approved it for that, then there could be some ability to treat similar diseases but different genes.
That would be the idea, I think, for say, an AAV gene therapy.
Yep.
But I think for genome editing, you could almost go farther than that. If you had a delivery system for the CNS and an editor for the CNS, you could use that same pair over and over again, and you'd simply, and I say simply in quotation marks because I know it's not simple, but you could just simply swap the guide RNA sequence, and the regulatory focus could be on that. And I think if we did it that way, that would be a way to develop this as a therapeutic platform that would make a lot more sense and treat a lot more people.
So we've talked a lot about AAV. We've talked a little bit about lenti cell processing. I mean, when I started, it was 10 stacks and mesenchymal stem cells years ago. I had no idea what AAV was back then, 20 years ago. So maybe the question that I have, we're all in the world that we're working on. What is it out there, out the window, innovative research that someone else is doing that gets you excited? Something in, let's say, base editing or different types of gene editing. What's new that recently been published that you think, you know, this could address problems that we can address? Let's say, like everyone knows that there's a packaging capacity in AAV with 4.6 kbp. Lenti can do slightly larger genes. But there are some very large genes that are associated with other rare diseases that, you know, are there technologies that you've seen that can package large genes, or what else do you see that's on the horizon, even within 5 or 10 years, that gets you excited?
Well, I can start. There's a lot going on, right? So people are doing things in many different ways. Transposons, you know, trying to find gene editing, trying to find templates, whether AAV or something else, to do specific gene insertions. So in my mind, the reality is we started with low-hanging fruit, a vehicle that was evolved to basically bring genes, is the viruses, right? So that provided the vehicle and the gene package, right? Like it was the perfect solution. We take the viral genes, we put our gene, that's it. But I do think that eventually we'll go a little bit more synthetic, based on
Synthetic
COVID,
instead of viral.
Everything, like in my mind, biological systems, you know, have their limitations. And, you know, there's a lot of things that we can do in vitro now, synthetically, plasmids, LNPs, you know, other ways of delivery that are going to be less complex, more defined, and potentially become more effective than the viruses, but we'll see.
Yeah, I guess maybe adding on that a little bit, and a little bit in danger of overusing the AI word, a poorly understood technology that's kind of looking for a home. But I do, you know, building on that idea of synthetic or unique and novel systems, you know, whether we're talking about designing a guide for editing or some sort of protein structure for delivery, you know, I think leveraging that as a tool to understand the systems to then build a better system. It feels like we're on a cusp there. I think there is a little bit of danger of either overusing that or misunderstanding how that could be a tool. And I'll kind of nod to my colleague here on my right to maybe educate me more on that space. But I do think that, you know, we've got an opportunity to look at big data sets, and to, you know, from a CMC guy, we've been collecting pH and CO2 and all of these variables for years and years and years, and everybody kind of, at least in my experience, sort of looks at them, says, huh, and then throws them away. But now we've got the opportunity not only to collect that, but to try to understand it, and try to understand that, again, to understand a system, to then use that to build a new system or to push a system somewhere where we didn't think it was going, or isn't out there right now. You know, it isn't naturally occurring, but is something that is wholly synthetic.
Yeah, for sure. I mean, I would echo that I think we are on the cusp of using AI for biology. And if you think about ChatGPT, right, it was trained on the whole of the internet. And if you have a foundation model that comes from metagenomic assets and just sequences of things paired with well-labeled data, like the measurements that you do, then you can really harness that for task-specific learning. Like, tell me a protein or a piece of DNA that will have a particular function. And my understanding is that up till now, the use of AI in therapeutics has really been limited to small molecules because of our ability to have synthetic chemistry, right? And now that we have these large gene sets to train models and then collect all this data, then we can start to unlock the same methods, but for these are like protein and molecule level questions. So that's something we're pretty excited about at Ginkgo. We've been our whole platform is based on automation and high throughput experimentation, and so we've already been collecting that kind of well-labeled data for years now, and then together with our new partnership with Google Cloud, we'll be able to better harness the compute power and with their help build these foundational models that will then go out to all of you for your products. So that's something that we're definitely excited about. And I wanted to come back to something you mentioned earlier about sort of, I think the question was around like the inefficiencies in industry, and something from where I sit in partnering with lots of companies is I just hear people coming with the same problems every time, and they're trying to solve them all themselves. And so it seems like some kind of standardization or outsourcing to really allow the therapeutic developers to focus on their special sauce. Like, what are you putting in your AAV? What problem are you trying to solve with it? Maybe, we shouldn't have 10 people trying to solve the AAV challenge. Just give that to somebody else, maybe Ginkgo, and focus on the actual disease or the payload or the special sauce that you really are the experts in. So that's something that I've just noticed from the partnering role that I have that seems like everyone's trying to reinvent the wheel. And so if there is a way to standardize on whether it's the analytics or the actual outsourcing of the work, it seems like it could help accelerate the product development.
Yeah, I guess just to pick up on that, one of the other things we're doing at NIH, the Platform Vector Gene Therapy Program, is as we're taking 4 AAV gene therapies to the FDA and ultimately we hope into the clinic, the things we learn from those interactions with the FDA, we're making all that information public, including, for example, how to submit an orphan drug designation. We have one. We got it, we made it, we have a template that we made public, and to try to do that, to make as public as possible whatever information we can, realizing that some of it is, of course, proprietary. But I guess just in terms of looking forward, I think I've been completely blown away with first the base editor, but also the prime editor developed in David Liu's lab and the continued progress in that area. I think the technology for genome editing is moving so fast. We have the technologies now, I think, to treat many, many diseases. The problem is not so much innovation there, it's more innovation in the regulatory space, the way we think of the process space, and also, as I mentioned, in the delivery. And so one of the most intriguing things to me is to take one of the few benefits of the COVID pandemic, the realization that you can deliver messenger RNAs in nanoparticles That was the COVID vaccine that was recently the subject of the Nobel Prize. And if we could take those messenger RNAs and instead deliver genome editors and guide RNAs and target those, that's an approach that I think has very broad implications, and that also avoids this sort of gene too big problem, because you're just delivering the messenger RNA of the base editor or prime editor.
So you're all affiliated right now with organizations in the U.S. Some of you've done research overseas. Do you see innovation, let's say in Europe or Asia or other places, in certain fields that you don't see here? Is there, let's just take a global view on this. Are you seeing any new pockets of innovation in other parts of the world in cell and gene therapy?
I can take this. I don't have a clear answer for you, but I think process-wise and analytics-wise, CMC-wise, I would think it's very, very similar across the globe.
Okay.
I haven't seen major differences. Everyone seems to go, okay, adherence system, suspension system, let's investigate producer cell line for process intensification. Everyone's trying to find better non-viral delivery systems. Cell therapies, same things, closing the system, automating, shortening processes, better understanding the phenotype of cells. But yeah, when we talk about cell and gene therapy, it's not a therapy. There's so many different therapies, and I don't think there's going to be a solution for all the therapies. It's going to be many different flavors for a while until we find, what fits better for each one of these types of indications. I do think that eventually, once we have enough understanding, we'll be able to skip the whole thing and go directly in vivo, right? And it will become very simple to do gene modifications in vivo, and we'll have a better control. But I think we need to take the time to invest, to understand the processes that we call platforms, understand them well, understand the products well, how the attributes correlate with clinical efficacy, and learn more, and then eventually we'll make great progress.
Well, not necessarily a international development. I do think that there's a role for the academic-sponsored clinical trials, because they can seem to get new medicines into patients faster. And at the end of the day, it's that human data that will drive new technologies forward. And so it seems that that's a route where, taking away maybe they might not be innovating in process, but in trying new things in their patients, it seems like that's a faster route to testing new therapies that I think, for me, has been really, illuminating to see those data streams coming out faster than, companies that are trying to build everything from scratch, I guess.
Data streams from different academic groups across the globe.
Well, not necessarily across the globe, just in general. Academic, clinically sponsored clinical trials seem to generate data faster.
Yeah, maybe just to follow up on that, I do think, in different countries there are different regulatory agencies, and they see things in different ways, and I think more sort of convergence around that, because sometimes they are able to move faster in some countries than the U.S., but not always. So I think there's real opportunity there to have sort of a convergence of regulatory paths, so that when something is successful in one country, it can benefit many others.
Any lessons or words of advice you want to give to members of the audience, things that you've lived through that you may want to share?
Yeah, I'd say, in my mind, people go for one or the other approach, trying to go very fast to clinic or, fast to commercial, right? I don't think you can have both, fast to clinic and fast to commercial. At one point, you have to take the time to really characterize, understand your process, control your product, understand your product. So I don't know if it makes more sense if you have a platform of products that are very similar to first do that piece and then kind of go advance the subsequent, once you have the manufacturing nailed down and you invested a little bit more upfront in that manufacturing, those assets and the understanding, then you can move targets really quickly. But if you try to move several targets all together at the same time, it's challenging. So yeah, investing in quasi-platforms, and going more fast to commercial than fast to clinic makes sense.
Yeah, I guess any advice that I would give is really a reminder that it takes a village to get a product out, even to get a product into kind of a process development or CMC pathway. It takes a lot of work from a lot of people. I think one of the things I saw as I transitioned from sort of standard biologics into cell therapy and then into gene therapy is that, again, in the CMC space, we talked about breaking down silos. We talked about how we all wanted to work together, but we were all so conditioned to work in, as we said, upstream, downstream, analytical. You had these very discrete units around a technology. But when working in a novel space and a novel modality, you become very aware very quickly that any change you make in any one of those is going to impact the other ones. All of a sudden you change something in your upstream process and you're making more material, you're putting more material onto then a downstream column, you're blowing out your analytical ranges and you have to reevaluate anything. And everybody's trying to make a decision right now. And so if you're working in those kind of established technology silos, it really can send you down in a tailspin of thinking you made a choice, but you didn't understand it, and now you have to go back and reevaluate it, and it just gets lengthy and confusing. People start pointing fingers, and it can be very, very frustrating. And so, I'm a big fan of running a lean team. My team probably hates me for that, but it does mean that we can all be in a room, and I force them to do these data dumps and really talk through what we're seeing and why we're seeing it. And it takes time because the vocabulary across those different unit operations is so different that people have to learn how to talk about,
It's whole new languages, yeah.
Yeah. Cell productivity to a downstream person it's just like, holy smokes, we all have to learn. But yeah, I guess the piece of advice is, keep your team matrixed, make sure that they're talking often, that folks understand the experiments that are going on, why they're being involved in that, so that you can all learn together, because otherwise it takes a long time to dig out from some of those confusions or mistaken experiments.
Well, not quite advice, more of an anecdote around the importance of analytics, and especially for new technologies. I used to work at a Cas9 company developing allogeneic cell products, and this is when Cas9 was pretty new, and the method for assessing off-target editing, though it was also new, and every company was saying, oh, I use CiteSeq, I use GuideSeq, I use CircleSeq, and there was no standardization around that, and the FDA didn't know how to handle all these different results coming from different assays. And so maybe an alternative approach that I think what was taken was a consortium from the leading companies working with NIST to say, okay, let's do all these assays head to head and compare and have sort of a, we don't want one company's assay to win over the other, but we want to advance all of our products forward. And so giving the FDA kind of like a standard way to think about a new technology that'll benefit all of the products in that modality or in that field from that one anecdote seemed useful. And now I think the question of off-target editing is largely solved. You know, the differences between the different methods I think are well understood. And it's not like you have to do it a certain way. The FDA is familiar with them now. But at the time it was a big problem, and there was a real threat that we're gonna have to use an assay that we don't like or that we don't ever do because that's what the FDA wants. But it could have been solved, you know, could have headed that off earlier on. And so maybe that's, you know, a cautionary tale for those developing new treatment modalities to also think about the analytics and how you're going to prove to the FDA that it is safe.
Yeah, I really like that. I think that kind of leads into what I was going to say, is just thinking about dealing with the FDA, and the very, very limited experience I've had is I do think some of these technologies now are really, really new, and they're really, really new to the FDA, and you have to bring them the, you know, solid scientific data. And I think to not assume that they're going to react a certain way. I often hear people sometimes in companies think, oh, we could do it this way, but the FDA is going to hate that, or the FDA would never allow that. And I think sometimes it's worth just asking them, you know, bring the data, bring the scientific data, make your case, and you might be surprised. I don't work for the FDA, so just be clear, I'm not speaking for them. But that's my experience.
So I think we've covered a lot of topics. Are there any topics that any of you wanted to cover here on innovation, or what do you think over the next several years? Anything that I didn't touch upon? I don't know if we can take any questions from the audience, but
yeah,
we still have 5 minutes. We're more than happy to do so.
All right, I'll ask. John Zalganis. I'm relatively new to the industry, and we're talking about standardization, and I'm very surprised that we aren't talking. And I know there's issues with money and people's IP, things of that nature, but it just seems to me like the simple answer is to get that group together, and there's a lot of base knowledge, and you've all touched on it. What is stopping us?
Yeah, I think, you know, historically, as companies, we play it very close to the vest. You know, if you've got a competitive advantage, you want to use it. We're all trying to stay companies one more day, you know, and that it hopefully doesn't become aggressive across companies, but certainly we're trying to live. I do believe that in the space that we're in cell therapy, gene therapy, where the patient populations are smaller, the therapeutics are more bespoke, we have to figure out a way to share information, because we will never generate enough data internally to understand what we're making. And that's, you know, kind of broad claim, but until we can, and I think it was mentioned, having these consortiums come together to understand what is the bare minimum CQAs in any modality that you need to understand to be able to put into a patient. That's not a company problem. That's a you know, across the industry. So all of a sudden, that isn't, I don't necessarily call that a competitive advantage, because we're, you know, as some have said, potentially a couple of safety events away of having the industry sort of fizzle out, and nobody wants that. You know, that's bad for everybody's business. That would be my 2 cents there.
And I just say, I think that's what we are trying to do in the Bespoke Gene Therapy Consortium, and I give great credit to Peter Marks and his role in that, because I think that's what brought these big companies together with the NIH. I mean, big companies don't need NIH grants, right? But the Venn diagram of interest we all have is to make the process better. And we can share data and know-how within that consortium, but I think part of the trick to it is we're developing AAV gene therapies for multiple diseases, all of which are so rare they're of no commercial interest. So we've taken the commercial interest out of it, and therefore we can have some conversations that will ultimately benefit, I think, the whole field.
In my mind, the manufacturing platforms, they all still have their flaws. So I don't think the government is going to say, use this for your CAR T in this process, right? Or use this assay. They're still trying to understand what's the best solution, and they're letting everyone invest and figure it out, But, we're going to learn over time.
So, question?
Yeah, so I just wanted to offer some joy from this. If anybody has ideas about other consortia that you're interested in, I mean, we're always happy. We run a lot of workshops. We've got like these 3 or 4 huge consortia right now that are very effective. And it is a great, I mean, this is the only way you can do this, and especially for sharing data, critical.
And maybe that's making a plug for ARM. You know what? I know that we were involved on NIST headquarters, I think it was, back maybe a year ago now on characterizing gene therapy, AAV gene therapies. And so I think, this organizations and others like it, allowing us as industry to come and present our data in a data sharing fashion really does help push that forward and answer those questions of like, what is holding us back? Why are we not sharing when we've got these big challenges to get through?
Might have another minute or two if anyone else has another question. There's one in the back.
Bring up a lot of good points, but when you look at the practicality, if you have a new set of modalities, you have new leadership, new people at the FDA, how do you choose your battles and find out whether or not, how when you have a group that really doesn't understand what they're managing because they're all new, how do you go to management with good faith and say, I've got this great idea to maximize this, this, and this, and be able to extrapolate how that's going to also deal with comparability issues that are just taking the industry and strangling it.
So, I can give Rocket's perspective. We don't look at it as going to a battle with the agency. It's actually been very collaborative. We have a lot of respect for Peter Marks and his group. And we announced, I think, 3 or 4 weeks ago, alignment on a registration trial for Danon disease. And it's a single-arm study in 12 patients. But we'd been communicating with the agency for several years now. And the discussion was very collaborative. There was a lot of additional information that was sent over a period of months. And there's mutual respect on both parties. So some of this is...
My point was earlier stage.
Yeah.
Trying to develop a strategy. Really getting out of phase 1, going into phase 2, not later phase stuff. Okay? Not the later stage stuff. You've been doing that for years, but you got all these new compounds, all this new stuff that you can, all new tools, new platforms. How do you choose which one? I don't want to use the word battles, but how do you choose which ones you want to take forward based on some of those experiences and successes you've had to getting things, let's say, beyond phase 2, phase 3? How do you do that?
I think each company has their own internal go, no-go, and a very high bar for science. You go back to...
And you just talked about getting together and trying to share some of this best practice stuff. This is the forum where people want to hear answers to some of these questions.
So the question is, how do you select the platform to manufacture your product?
How do you make these based on your experience and how things, the industry, your diagnostic capability, all this is growing up. And so what you did 3 years ago, you have all these different experiences and you go to management. This is going to take a little less time. We've got all these issues, but I feel very confident because of X, Y, and Z.
Yeah. So I think when people make those decisions are a combination of several factors. It could have to do with capabilities that a company already has, expertise the company has, information that is out there that shows that that type of manufacturing process will deliver a safer, more effective therapeutic, and also IP. So I don't think it's a one, the decision on how to manufacture depends on one thing. It's a combination of multiple things. And they know one way people are developing therapeutics. Every single one is developing slightly different. So we're going to see. I think we're learning. We're still in the learning phase. we're not ready to lock.
Yeah, I guess maybe just adding on that, I've found that data is the most important thing that you can walk into in that conversation. It's great to have high aspirations, it's great to have hopes and faith, but without data, it's always going to be sort of this question of, is this going to work? Is this the right time? Is it not the right time? And I think probably all of us on the stage have had to cajole or twist arms to get resources to do one experiment to really, whatever that capstone is, to convince somebody. But I think that is key, is showing up with the data.
Yeah, and maybe just to add, I don't think people are spending as much time thinking about the manufacturing at this point. It's really, they have something, they've shown preclinical data. Usually we're targeting some sort of unmet need. We want to see proof of concept in the clinic before, a lot of people just really go for the fast to clinic, and then they revisit their manufacturing strategy, which ultimately takes more time to go to commercial, but that's what's happening.
Thank you, everyone, today. So it was a great panel, and looks like we're over time.